Okay, I'm gonna hit record.
Close.
Okay. So, welcome everybody to our February Science on Tap with Jessie Lopez. And we have some people in our audience that don't usually attend Science on Tap. So to set the stage, we are in beautiful Santa Cruz, California. And in Santa Cruz, we usually have Science on Tap at somewhere called the Crate Place. So I want you to imagine yourself at the Crate Place, sitting in the back, maybe sipping on a beer, waiting for some snacks as the speaker gets ready to share some science with you. And a little bit about our presenter, Jessie. Jessie was a special education assistant at Los Angeles Public Schools for 14 years, where she began to explain things simply and creatively. And then she attended community college for 10 years and she studied things like art, painting, drawing, music, acting, physics, ecology, creative writing, chemistry, among many other interesting topics. So she knows so many different things. And then eventually, Jessie was at a crossroads where she either was going to continue education to become an animator or a scientist. And while at this point, Jessie is currently getting her PhD at UC Santa Cruz studying the spliceosome, she maintains that vibrant creativity that will shine throughout her presentation. And as Jessie mentioned earlier, if you have any questions, please feel free to unmute yourself or just put it in the chat and I'll be happy to read those aloud for you. And with that, Jessie, please take it away.
Yay, thanks, Megan. All right. So it would be nice if I could do this in non-Rona times at the Crate Place because it's a nice spot. But I'm so happy to be able to do it now. And I love that people can come from all over the country. And shout out in particular to people who are on the East Coast because I know that this is late for y'all. You have a baby. So, when you're watching a TV show or playing a game, let's say you're reading a really good book and you kind of get immersed in this other world. You're in West and Hogwarts or whatever. Well, for the past 5 to 8 years, I have been spending a lot of my time immersed in the world of molecular machines. So these are very, very small. And it's weird down there. It's very, very different from the world that you and I normally live in. And so I'm going to part of why I'm really excited to do this, and I'm so grateful for this opportunity, is because I want to tell. This is like when you're telling your grandma about playing Pokemon so that she understands what you're into. I want to tell all my friends and family about where I spend a lot of my time, and I spend a lot of my time imagining inside the world of molecular things. So first, I'm going to tell you about molecular machines and the world they live in. I'm going to tell you about my favorite machine, the Spliceosome. When I give talks to other molecular biologists, they're often like, oh, the spliceosome, because it's complicated. But hopefully, I'm gonna make it simple so that you can see what I think is so amazing and beautiful about this machine. I'm gonna set up what I've attempted to do in my science, and then finally, I'm gonna tell you a little bit about what I've found. I'm not gonna give you the whole story because it gets more complicated, but I've got some exciting results, and I'm so happy to share them with you. And as Megan mentioned, if you have questions, put them in the chat, or I just interrupt, I guess. So you are huge. People often look out at the night sky and look at this little tiny rock that we're on and the immensity of space and say, wow, I'm so small. And that's true. But on the other side of things, you are also gigantic. You are made of cells, and a lot of those, right? So if you think of 1,000,000 cells, that seems like kind of a lot, right? Double that, you get to 2,000,000, 3,000,000, 4,000,000, 5,000,000. When you have 1,000,000,000 cells, that's the range, okay? You have a lot of cells. And that's the actual unit of life. That's how your body actually operates. But you're so big, we are so big that we can't even see ourselves anymore. We can't even see inside of ourselves to see how those little units work. Inside of each of those cells are molecular machines. And just the same way that your body runs is the work of these cells creates you, the work of these molecular machines creates the cell. So if you take that trillion molecular machines, once you have a million trillions, that's how many of these molecular machines are in every single one of your cells all the time, working hard, keeping you alive. That's a lot. So we're going to dive inside and see a little bit about how things are on the inside. So here's a protein. This is a molecular machine, and it has a shape. It is a physical shape. It is an object in space. It's not really these colors. Those are false colors. And it also doesn't really have hard limits because each of these little spheres is, you're looking at atoms. And so atoms are much weirder than, they don't act necessarily like things on our scale, but we can totally imagine them as spheres, hard spheres, and that gets us towards understanding a lot of their behavior, at least at the scale that I study. Proteins have a sequence. They are made out of subunits, and so it's actually a long chain. So each protein is one long chain, and it's, here's like one of them. You can see I outlined it in white on the protein. There's another one. There's another one. And that long chain gets folded up into the shape. Sometimes it folds up on its own. Sometimes it needs help. We could look at every single atom, but that's awful. I don't want to look at that. What I do more often, what most molecular biologists do more often, is we look at ribbon models. And so you can see there's these kind of coils. Those are alpha helices. And proteins have structures, and they are common, and they help us understand how they work. So you see these things over and over again. So a beta solenoid, this alpha beta 4-layer sandwich, we give them names that kind of help us make sense of them. And we see them again and again. So the same way that you might see a new animal that you've never heard of, and you can recognize parts. There's the nose, eyes, ears, tail, right? Because you've seen a lot of those before. And so you have some idea, so you go, okay, it's a beta barrel. So I know certain things about it, right? And it's really good that they have these things in common because that helps us. So for instance, the spike protein on this bat coronavirus has been studied for years, which is why when SARS-CoV-2 came along, we already had a head start on understanding this spike protein because they are so similar. And they're similar because they have a common origin. And this talk isn't really about evolution, but there's beautiful, beautiful things to learn about evolution from looking at what they have in common. So, okay, so let's look at one of these. For a protein, for a molecular machine, gravity, momentum, not really important. Okay, gravity and momentum both are based off of mass, like mass is part of it. And these things are so small, their mass is so little that gravity and momentum aren't really big forces. For us, they are. For proteins, they're not. Physical shape, that still matters, right? If you can't reach it, then you can't reach it. So if there's two things that have to touch and one of them is, back here, then they can't touch anymore. What matters for molecular machines, things like positive and negative charges. If you imagine two ends of a magnet snapping together, a positive and a negative charged part of a protein can snap together and hold on to each other just through virtue of their positive and negative charges. pH, which is the number of free protons that are around, that's super important. There's a very narrow range of pH that these operate at. And if you go higher or lower, they don't work. So a lot of the alkalizing products that are out there being sold are because if you change your pH too much, your proteins won't work. Van der Waals forces, this is like two non-polar things that kind of vibe together, and hydrogen bonding, which is polar things that will make little bonds. Those two forces, van der Waals and hydrogen bonding, they're small. But if you put enough of them together, then they're really powerful. So if you think of one tooth of a zipper, it's not all that strong, but you put a bunch of them together and you can really close up a space. And so the van der Waals and hydrogen bonding are kind of like that. The most powerful force in here is hydrophobicity in terms of physical forces. And the reason for that is that all of this is happening in a background of water. The chemistry of life is water chemistry. And so on our scale, hydrophobicity is not that big a deal. It's, you know, you got to mix your salad dressing because the oil and water have separated. But in the world of very small things here, if you have two hydrophobic surfaces, two oily surfaces, they don't want to interact with water, and so they will click together and hide their faces next to each other so that they don't interact with the polar environment for reasons that I don't understand, but I use all the time. So your living bodies, our living bodies, are the result of constant work of molecular machines. And I know we're deep in the rabbit hole now. It may seem like this doesn't really have a lot of relevance, but this has definite immediate relevance all the time. So this is an ATP synthase. Perhaps you've heard that mitochondria is the powerhouse of the cell. Well, ATP synthase is why mitochondria is the powerhouse of the cell. You have, I didn't do the back of the envelope calculation to figure out how many of these you have, but you have a lot of them and they're working all the time. And in fact, this is the reason that a human being has to breathe oxygen. This is why we have to breathe oxygen. Because if you don't breathe oxygen and all of the ATP synthases in the body pause for too long, then that's not supportive of life. And we can't survive it if it pauses for too long. So there is a direct relevance back and forth. So I want to tour a little deeper into the world of the cell. And this is, maybe it's going to be a little chaotic, but stay with me. This will be the only video like this. So here we are inside of a cell. And there's water everywhere, but this video, the water is not shown. Oh, I need some water. What we're seeing is all of these proteins, and it looks very chaotic. Here, this rainbow protein is getting recycled by this kind of yellow one here. And you can see it's chewing it up into those subunits. And those will get used again. Those will get put into another one. This blue thing is a bag of maybe neurotransmitters. And all those yellow things on the outside are proteins that have to go somewhere. Well, how are they going to get there? Well, this little elbow that's opening up here, this is a molecular machine. This is a particularly charismatic one called kinase. And what this does is it takes these little steps along a microtubule, and it's slow. This is just not how things work on our scale. On our scale, there's a deliberateness to our actions, and that's not how it is for these things. They move kind of randomly, and then they bump into each other and they click together. But that little kinase can walk that bag of neurotransmitters all the way out the cell. Here are these green things. These are clathrins, and they are working together to pinch off a little ball of stuff here. And they recruit each other, they work together, and then once they've got it off, their job is done. And they drift away to go do other clathrin things that I don't know what they do. So in this is the environment. This is where I'm spending my time. Okay, this is the wonderland where I spend a lot of my time. And it's a little chaotic. It's a little overwhelming. And you may be wondering, how does this work? How can you possibly have all of these machines in this chaotic environment just bumbling around, bumping into things and finding each other? How is that going to keep a cell alive? How is that going to keep me alive for decades and decades as I go through things and change? So let's go somewhere less chaotic. We'll go to this library. Okay. So I'm in the nucleus. So in here, in the nucleus of the cell is the entire genomic library containing all of your DNA. So all of those trillions of cells that you have, each one of them has one of these. And in that library is all of the instructions to make, repair, manage, and recycle all of those proteins that we're looking at. It governs all their behavior. So let's say we want to look at one. We want to look at these instructions. We say this one here. Can we look at it? Well, no, we can't. Okay, there's ropes. This is restricted. This the genomic DNA is ancient. It has been traveling from body to body to body through all of these offspring for millions, billions of years. Okay, you can't just go in there and start mucking about with it. But this helpful librarian robot will make you a copy. Okay, so the information that is stored in the permanent collection, which is the nucleus, all the DNA in the nucleus, if the cell wants to use any of it, which it has to do constantly, the first thing that it does is it gets this mRNA copy, temporary copy, and then it leaves the nucleus and it goes to this other helpful robot, the ribosome, which is a 3D printer. And so it brings that mRNA, it feeds in the sequence, which is a long chain, and the ribosome reads it in triplicate. It looks at every 3 letters in the code, and then it matches the corresponding subunit of the protein. It 3D prints the protein and then starts making lots of those. You gave it the mRNA, and now it can start pouring out little micro machines to go do whatever they do. So just to summarize, DNA is the permanent record of the instructions to make and operate all of those molecular machines. Here's our friendly library robot who's making you a copy. The mRNA is the active instructions. It's the active form of that information. And then that will be used to make proteins, which is what molecular machines are made out of in general, for the most part. There's an extra complication with these mRNA active instructions, and I will tell you about it later. But right now, does anyone have any questions? If there's anyone, Megan, you can let me know if there are any questions. Okay, all right, so I'm gonna keep going. So how do we study these things? They are so small. What can we possibly do? Well, there's a bunch of techniques. So a lot of what we do is what I call Ewok science. Okay, a lot of these things are what I call Ewok science. Meaning, yes, I mean Ewoks. Meaning that in a way, it's kind of very simple and primitive. So if an Ewok, I'm just going to assume that there's eucalyptus on Endor. So eucalyptus makes compounds to combat bacteria, right? So it makes 1,8-cinnamol and enantiomers of alpha-pinene. If this Ewok grinds up those leaves, extracts out these compounds, and bottles them and uses it to combat toenail fungus, yeah, that's sophisticated, but kind of not. They're just taking something that evolved in nature to combat bacteria and fungus, and they're just putting it somewhere else to combat bacteria and fungus, right? So this is what I mean when I say Ewok science. You're literally just purifying something from nature and putting it somewhere else, okay? So one of the things that the first thing that people think of when they think of molecular, think of laboratory techniques, is a lot of times they think about microscopy, using microscopes. And for a lot of things, we can do that, right? We can look at an ant under a microscope. We can look at, a mammalian cell under a microscope. But when you get to things about this small, where this arrow is, it doesn't really work anymore, because once you're smaller than that, the light that you use to shine on something is so big that it smashes through the thing you're trying to study, and it doesn't bounce back and give you information. Okay, this is the world of very small things down there. So we can't really, there are some specialized microscopes that you can use to study proteins and small molecules, but in general, that's not how we get most of our information about molecular machines. I'm just going to make a point right now, just because my family's here and I would love for them to know this, that microbiologists study bacteria and also viruses. And so this is kind of the area. You can, if anyone wants to complain about this range that I put in, that's fine. But in general, microbiologists study things that you can see under a microscope, okay? Microscope, microbiology. Molecular biologists is studying much smaller things. They're so much smaller that you cannot really use a microscope as your main way of looking at them. So what I do is I'm a molecular biologist. So It's very common to hear people say I'm a microbiologist and I love microbiology. I mean, it's amazing. I need the results that they're getting. It's just not what I'm doing. So that's my little plug. Okay. Okay, so fluorescence is definitely Ewok science. Okay. So this is a beautiful rendering of a protein that was originally found in jellyfish. And now we have this whole crayon box of proteins. And in jellyfish, they have a gene. The gene makes a protein, and that protein, when it folds up into shape, if it gets hit with blue light, it glows back green light. Well, we scientists have taken something that evolved in nature and just put it somewhere else. And where we've put it is all over the place. So any protein that I'm studying, well, with some exceptions, in general, if I'm studying a protein, I can't see the protein. But what I can do is attach a little headlamp on it, right? So if I can add the DNA that codes for this protein to the end of my protein, then when the 3D printer prints out the protein, the whole thing comes out, and at the very end, it prints out this little lantern that bobs along wherever my protein goes, and I can see it through the microscope, and so it can tell me where my protein is, and that's super helpful. And so some of these fluorophores we've gotten from jellyfish, from lightning bugs, from sea pansies, and from all over the animal kingdom. There are so many different glowing things found in nature. And so we've done this kind of very simple thing of take it and put it over here, use it somewhere else. We do a lot of spinning things in the lab. If I have a tube with a bunch of different size, tiny things in there, if I just spin it around really, really fast, the heavy things will go to the bottom and the lighter things will be on the top, and then I can separate them out that way. So this is, I mean, it's, you know, this is merry-go-round science, but it's a super useful technique. Okay, so this one, I have to roast my mom a little bit on this because I explained PCR to my mom one time, and she got it. And then the next time I talked to her, she said, well, I just think that your discovery about PCR is so amazing. And I said, Mom, I didn't discover PCR. And she said, well, I think you're underselling yourself because I think it's amazing. And I said, Mom, everybody does PCR. And she said, I don't know anybody who does it. I think it's amazing. I did not invent PCR, but actually it's a cool story. I think that the guy that came up with it was on acid at the time when he thought of it. Anyway, PCR is definitely Ewok science, according to my definition. So at cooler temperatures, this protein, this polymerase, is inactive. And if you warm it up, suddenly it picks up and makes a copy of whatever DNA is nearby and copyable. So in the laboratory, we have a little machine on the desk, on the bench top. It's like, you know, smaller than a microwave. I put some little things in a tube. I put some DNA in the tube. I add this protein and press some buttons, beep, boop, boop, and I go to lunch. And then when I come back, this lovely little machine has made 1 billion copies of the DNA that I put in there. So this is not the whole library of everything. This is one page copied over and over and over and over again. So many copies that I can actually physically see them in, you know, on our scale, which tells you that's a lot of copies because these things are small. And then a technique that goes along with that is enzyme digestion. And this, I always thought this name was so weird when I was first learning about this. So this comes from bacteria. There's lots of different ones of these. And basically what these enzymes are is it's a protein. Here it's in a red, white, and blue. And what this protein does is it finds a particular sequence of DNA and it cuts it. So that protein looks for somewhere that it says GAA-TTC. Oh, I forgot to mention, DNA always runs anti-parallel, which sounds confusing until you realize that like freeways also run anti-parallel, streets run anti-parallel. Yeah, you got northbound, you got southbound. It's not a big deal. Anyway, so the DNA, when it finds that, it cuts and it leaves this little, you know, you can see the second picture there. Can Can you guys see my mouse? I don't know if you can see my mouse.
Yeah.
Okay, yeah, thank you. So down here, you can see the cut version of it. Well, that was cool when this was discovered. So what can you do with that? Well, part of what we do is we take that, and let's say I have some copies of DNA that were made by my trusty thermocycler, and I have a mutant DNA. I add some of this protein, and then I see if it cuts them. Oh, that other one totally got cut. Okay, so what that tells me is that that has the sequence that is cuttable, and the other one doesn't have the sequence that is cuttable by this enzyme, right? So that's super useful because these piles of DNA are so big that I can actually see it on a gel. And that's the next technique I'm going to tell you about is gel electrophoresis. So this is another thing that sits on the benchtop in the laboratory. We have them all over the place. If you went to like a good high school, maybe you did this in high school. I didn't, so I didn't do this until I got to college. And it's this like not disc because that's round. It's this flat rectangle made out of like fancy jello kind of. And it's got these little holes at the top. Those are the sample wells. And you take a little, you squeeze in your samples up at the top. And what it is it's this tangled matrix of gel. And so all of your little particles are up at the top. And then you flip on that power supply, and current pulls things down through the matrix of gel. The little things can slip through faster. The big things, it's harder for them to wind their way through the tangle, and so they're slower. And so just like the centrifugation separates things, gel electrophoresis separates things. And what you wind up with is something that looks kind of like this. And the longer bands of DNA are at the top, the shorter bands are at the bottom. So for instance, so there's lane number 1, lane number 2, if in lane number 3, maybe the enzyme digested and cut that one, and that's why there's this little short piece at the bottom, right? Could be. Yeah. You can also do gel electrophoresis with proteins. You don't have to just do it with DNA. I don't do Western blots, or I haven't yet, but that's definitely a thing that I should probably learn, maybe this year, maybe once I get a vaccine. Okay, so in terms of genetic techniques, a really common thing that we do, this is not exactly Ewok science, this is Hulk science, okay? So if there's a gene that causes certain protein, and I'm trying to understand what that protein does, one of the options available to me is just to Hulk smash it. It's just to knock out that gene, which means that you don't have the protein. And then I go ahead and I look at the fly and I go, oh, it doesn't have any eyes anymore. And then I get to publish. Organ patterning affected by eyeless gene in Drosophila. This is not a real citation. This is a nod to Stanley. Yeah. Similar in terms of genetic techniques is overexpression. So here there's a gene, and let's say it makes something, and I go, what if there were a lot of these? What if this gene produced a whole lot more of these? And then I look at what happens, and then I get to publish. And so as much as these are like super sophisticated, these techniques are also, we're like, what if we smash it? What if there was more of it? And gradually, all of these scientists the world over doing these experiments and publishing them and putting them where everyone else can see them, we can start to piece together stories of what all of these molecular machines are doing and why they're doing it and what it all means in terms of life. So if you want to compare, so this is a technique that I definitely do all the time. So the last two, those are genetic techniques. Those are things that is part of how I personally work right now. I definitely do a lot of multiple sequence analysis. And so what this means is if I look at the sequence for a gene in one species, and then take another species and compare, line those up next to it, and the computer does that part for you. What we do is we look at where are they the same? Where are these two proteins exactly identical and where are they different? Because as the DNA gets passed from organism to organism to organism, it's true, it's super important. But, everybody makes mistakes. There's like a letter here, a word here. There's little mistakes constantly happening. And if those mistakes are too important, then the cell doesn't live, then the organism doesn't live. So eventually, what you have is this picture of what are the most important letters in the sequence, because those are the ones that have to be there, that have to stay the same, that are maintained over time. And I kind of think of it like if you were hiking in the forest and you're going past all these trees and brambles and streams, and you suddenly come across a rose garden in neat rows. Well, that is being maintained. There's no way that you have that rose garden just randomly out there. Um, the natural wild plants being born everywhere would have reclaimed that spot if there wasn't something keeping it that way. So when you have these parts of the protein that stay the same, it's kind of like that. It's like, this must be the important part, because as this DNA is moving through millions and millions of years, millions and millions of bodies, that part has to stay the same. So it's being maintained by evolution. And the way you can think about this, if you look at, the first time I ever saw one of these flash drives, I didn't know which necessarily is the important part of it, right? There's this cool slider, it's got a ring on it. I don't know, maybe that's important. But eventually, when you look at a whole bunch of flash drives, you realize that there's really a very small part of it that's the important thing. And the rest of it's kind of window dressing. Also, these all evolved from the same idea about how to store data. So there's a lot of And so the thing I want you to see about it is these little, this is the RNA, would be an exact copy of the DNA above it. But see these little boxes here? These are the pieces that are actually going to make it into the final mature active mRNA that can go on and make a protein. And all of those pieces in between those boxes, they're just in between, they're introns in between. Those are just going to get cut up and thrown away. Happens every single time. So this DNA has been traveling for billions of years through body after body, lifetime after lifetime, and every single time it gets made into an active form, the spliceosome looks at those introns, those in-between areas, and goes, nah, no, we don't use that. It's stupid, whatever, and throws it out. And it needs to do that to make this active information. But part of what we don't understand still is how it knows what parts to keep and what parts to throw away. Because if I just take the sequence that this shot is based on and scroll it, I don't know where to cut. Okay, there's a string of A's. Does that mean I cut there? Right? Like if I look at this, it all looks very confusing and it's not obvious to me where to cut. So I'll tell you what we do know about how the spliceosome decides how to cut out those introns and throw them away. Okay, so we know that in general, the 5' splice site, this is a little piece of jargon for you. I've mostly tried to avoid it, but this is jargon. 5' splice site is where it makes the first cut, where it's going to get rid of stuff. And then that happens in general right before a GU. So just, in that string of letters, when we look and figure out where the spliceosome cut, we see that it's always at a GU. Sometimes, less than 1% of the time, if there's not a GU, it can at a GC, but in general, it's a GU. The 3' splice site, 3' splice site, is where it cuts the other side, and that is right after an AG. And the 5' and 3', don't worry about them too much. I mean, on the freeway, that basically means like the on-ramp and the off-ramp, right? The 5' is just our way of saying this side of the double-stranded. Anyway, the thing that you need to know is the 5' is the beginning of the part that's going to be removed, and the 3' is the end of the part that's going to be removed, and they're going to get stuck together. So yeah, that GU is where it cuts. Every once in a while it's a GC, but that's less than 1% of the time. So if we pull up that sequence for that role 1 gene that we were just looking at, I'm going to go through the sequence, and everywhere there's a GU, I'm going to color it green, and everywhere there's an AG, I'm going to color it this magenta color. So before I do this, I want you to think about what are your expectations? Where are you expecting to see the GUs and the AGs that the spliceosome uses as landmarks to cut? So here it is. What? They're all over the place. I look at this and I'm like, well, this is a mess. How do I know it? How does the spliceosome know which GU to cut at? Because when we pile together all of the places where the spliceosome makes that 5' cut, the only thing that they all have in common is the GU. They don't have anything else in common. That 3rd, 4th, 5th, 6th one, it could be anything, but it has to be a GU there. Why? We don't know. We don't know what, there must be more information. There must be more information in the system somehow. So the project that I'm currently working on, that I'm going to tell you more about, the primary thing I'm looking at is the 5' splice site and how that's chosen. I don't know how I'm doing on time because I can't see any kind of clock. Okay, so this is the picture that we always use for the splicing cycle. Every splicing talk, it's required by law. It has to start with one of these. And I know it looks complicated, but I'm going to break it down for you. Okay, so you know how I said that the spliceosome, it's very dynamic. This is one big picture showing you, kind of summarizing all the changes that happen to the spliceosome. So as I walk you through this cycle, what I want you to keep an eye on is that 5' splice site, because it's going to get passed from hand to hand like a baton in a relay race. Okay, somebody's holding it, and then somebody else is holding it, and someone else is holding it. And so I want you to kind of keep an eye on these like escorts that have it and when they hand it off. You're not going to know, you don't need to know the details, but I just want you to see how often this is happening. So at the beginning of the splicing cycle, the first thing, one of the first things that happens is that the 5' splice site and the 3' splice site get identified by these molecular machines. And as the splicing cycle proceeds, things move around. This little trio of proteins comes in, or not just proteins, a molecular machine. And that initial thing that found the 5' splice site leaves and is gone. And now these three have it. And then you keep going through the cycle, and now one of those three leaves. And now these two have it. And you keep going through the splicing cycle, and the two ends are moving close to each other. And you keep going, and there's a bunch of rearrangements happening in the big machine around you. And all the way over here in the splicing cycle, you finally get the first snip, the first step of splicing. And that 5' splice site has been cut. And then you keep going, and the 3' gets cut, the two ends get joined together, the intron gets thrown away, and you finally have your mature mRNA. Just that one intron has been removed. You have to do this whole cycle again for each of the other parts that you're going to cut out. And you have to recycle all the pieces, because they're going to have to find the next intron and do this whole thing again. So, you know, I said that you just have this snapshot at any one time, because it's really the sequence, the whole thing, that creates the action that it does. And I know that these just look like spheres here, but each one of these is its own complex thing. Actually, these U1, U2, U4, U5, U6, another complication I didn't tell you is that sometimes molecular machines can be made not just of protein, but there can also be RNA in the machine. And so these things have RNA in them. So what I'm studying is this strange phenomenon of the 5' splice site that gets passed from hand to hand all the way around, and how in all of that 5' splice site is maintained through all of the rearrangements of the splicing cycle. Just like how, it's amazing. Yeah, so this is the spliceosome. So I've told you a little bit about how it works. And if there's any questions now, that would be a good time before I go on and tell you about my goal, my quest to understand how it thinks and how I'm going about it. Okay, I'm not hearing anything.
Hey, Jessie.
Yeah.
No questions in the chat, but just because you asked about time, it is about 8 p.m.
It's 8 p.m. Okay, cool. Yeah, that's okay. That's great. Okay, so thank you very much for letting me know that. I'm guessing I have around 20 minutes. It might be more like a half an hour. Yeah, so come and go as you please. Okay, so the laboratory that I work in now, and that I've been working in for a while now, we use this little nematode worm called Caenorhabditis elegans. Its friends know it as C. elegans. And these are hermaphrodites, so they can self-fertilize. They are found in rotting fruit. It's not this kind of worm, though. It's much smaller than that. If you lined up 10 of them, they would just cross your pinky nail. They're very, very small. If you put a pencil tip next to them, they would probably not like that very much. They have sensory organs. They have a little mouth. They have this grinder that beats all the time like a heart. They got, you know, guts where they digest their food. And they have this cool reproductive tract. It's got two sides, like kind of like fallopian tubes, and they fertilize themselves on each side. Each side works independently, and then they lay their eggs out their little vulva there, which begs the question, if a worm wore pants, would it wear them like that, or would it wear it like that? Because it kind of depends. what's more important for pants? Which thing do you got to cover up? So as small as these worms are, they have a DNA library. And actually about a third of that is identical to ours, because even though we have trillions of cells and they have about 1,000 cells, most of the instructions in the library are about running a cell. And so they're made out of cells also. They have spliceosomes, and I can use that CRISPR-Cas9 system in them. So I can design in my proteins and my guides and just inject it, ouch, right in the gonad. And I can just blow up a whole wing of that library if I want to. Or I can just change a single letter in a single book. So, okay, so that's cool. So what I need is a good example. Like, where's a good place where I could study how the spliceosome works? So we lucked out in our lab with this protein called UNC73. And if UNC73 gets messed up, then the worm, so what this gene does is it's a protein that helps the muscles talk to each other on either side of the body. And when it gets messed up, the worms look super duper messed up. And so we found this cool example, Al found this cool example, this happened years before I got there, of an intron in UNC73, and it has that normal GU dinucleotide, the little G and the U, and in wild type, oh, this is another piece of jargon. Okay, so this is a term that scientists use all the time. We have these laboratory strains that have been co-evolving with humans for a long time. We use them all the time. When we talk about wild type, what we mean is the wilder thing, right? We mean the more normal thing. So dogs and wolves. In our case, what we mean is a worm without anything weird done to it, right? We haven't done any genetics. It is a very laboratory strain, but it's just normal without any intervention on our part. So a normal worm, normal laboratory worm, is always going to splice at that GU dinucleotide, at that G. Sorry, I can hear myself going into my scientist jargon once I see these slides. So that's where it cuts. That's the 5' splice site. And this is a gel, just like the gels I showed you earlier, and it shows that length of DNA is what we made it into. It shows where it cuts. But when that G gets mutated to a U, we didn't do this, we found it like this, it makes a weird splice site. And what happens is that there are 3 splice sites that get used, because where it normally cuts, there's now a UU. Well, that's wrong. It still cuts there sometimes. Sometimes it goes back one and it cuts at this GU, this new GU, and sometimes it goes all the way over to this GU over here, because remember, there's GUs all over the place. And so it makes this these 2 new splice sites don't make a usable protein, right? They're not in the correct triplicate code. And so the result is that the worms that have just this one little U, that U instead of a G, these worms are dramatically messed up. About 75% of the time, a wild type spliceosome that sees this intron will cut at the +23, and that doesn't make a good protein. And then the rest of the time, it's split pretty even between this doublet of side-by-side splice sites over here, and only one of them makes a usable protein. And it's very sensitive. So I'm actually going to skip ahead a little bit and say that if we increase the splicing that happens at the wild type splice site, then the worms are suddenly happy again. So this is our exploitable situation here. This is our wonderful situation where we can figure out what's going on. So here I've got this worm, and its genetic library. So if I take a whole bunch of worms and hit them all with mutagen at the same time, zzz, right? What I just did was every single one of their libraries got randomly messed up somehow, right? So a letter changed here, a page ripped out there. Every single one of those libraries, something random different happened to them. And I take those worms and I put them on the edge of a plate, and they're hermaphrodites. And so they just self-fertilize and they lay eggs, and they can't move very well. So they just kind of roll over, and a couple generations go by, and I come back to the plate and I go, hey, that's suspicious. Why are you swimming so well? So what I can do is I can now look in their genetic library and see what changed. Something changed in their genetic library that allowed them to overcome this weird splice site and to move well. And so the analogy that I think of is, this made its rounds on the internet recently, about fighter planes that come back with bullet holes in them. If you look at where the bullet holes aren't, that tells you what are the most important parts of the airplane. That if they get a bullet there, the plane's not coming home. Okay, because when I look at a schematic of an engine, I can't tell what's important. And when I look at the spliceosome, I can't tell what's important. Even my car, I can't tell what's important. I don't know how it works. But if I shot the car full of very regular bullet holes, if I shot millions of cars with millions of bullet holes and I see which ones of them interrupted steering, eventually I would be able to map the whole steering column of the car by which of those mutations, which of those bullet holes messed up steering. And so that's what I'm doing with the spliceosome. I am messing up those genetic libraries, and I'm finding the places on the spliceosome where I can change what it does with this ambiguous splice site, so that I can map out the steering column, the decision making, the 5' splice site choice parts of the spliceosome. So that is the technique that I'm using, and that is how I made these discoveries. Everything else is following up on that. Okay, so I've told you this is my quest. I'm going to mess up a bunch of worms, and I'm going to try and understand the steering column. So what did we find? Well, we mutagenized, we messed up over 1 million worm genomes. We go through a lot of these, and we've been finding these little spots where we can change splicing at that spot. And we found so many that we started grouping them into classes. I think I'm going to try and speed up a little bit. So class 1 and 2. Class 1, these, we only have one example of this, and this happens really early in the splicing cycle. This is the first thing that comes in contact with the splice site. And I'm going to skip the little movie about it, and just say that in the wild type worm, the ratios that we see, I told you these before, about 78% of the time it splices at that plus 23, and then about 12% at the and 10% at the little doublet of splice sites off to the side. Well, when this U1 mutation happens, we see about 33% used each of those 3 splice sites. So it really, really changes the ratios. So we think that that class 1 is really about the initial recognition of the 5' splice site. Class 2, these are things that come on later in the splicing cycle. So they're not even present before that. You don't need to know what any of these are, but if you were a splicing person, you'd be like, those are good, PRP8, wow, that's legit. And what these class of suppressors do is we see this. So if this is how splicing looks like without these mutations, in these mutations, it looks pretty similar, right? But there's less splicing at the plus 23 and more at the doublet. And I'm also going to skip the movie about those. Okay, so the class 2, those shift splicing towards the doublet. Now, exciting. Okay, on to class 3. This is what my paper is about. This mysterious new class of splicing factors. So 2 proteins, PRCC and kin17. It's not a kinase, it's something else. Anyway, we have 4 mutations, 2 in PRCC, 2 in kin17, and both of them do the same super weird thing. So this is what splicing looks like in the presence of PRCC. It's changed the ratio between those doublets. it's not really drawing, it doesn't look like it's drawing too much away from the plus 23. It's really changing the doublet. And so if that's what, this is what wild type splicing looks like, this is what the ratio looks like in the case of PRCC. So you can see there's a whole, we've doubled the splicing at the wild type splice site. Kin17 looks the same kind of thing. We go up to 24, right? So this was our first glimpse of what these things did. And I'm going to save that off to the side over here so that you can see it later, because I'm going to tell you some more stuff about it. Super weird. What is happening at this doublet? So to study it further, we wanted to just focus on the doublet. And so we got rid of the plus 23. And in a wild type background, we see that the spliceosome is using it pretty close to evenly, a little bit more of the wild type. In the presence of our suppressors, again, it makes this shift. And now we see it's this like 70-30, and it looks the same in PRCC. So we're going to save that off to the side. Well, we wanted to focus more on this doublet thing, because this is what I was wondering. If the wild type site, the wild type splice site, which is normally a GU, is now this UU, when these mutations push splicing that way, are they going for the correct location, even though the sequence is wrong? Or are they for some reason going towards a UU? So I wanted to separate out sequence from location. And so this is what I did. I CRISPR'd this in. I took this entire region around the splice site and I doubled it, overwriting the downstream part. So this is a double. That's a California joke. It's a double. Okay, so this duplicated doublet. Now I have two of these little doublets. And in a wild type background, we didn't know it was going to happen. All 4 splice sites get used. So the original doublet gets used more. And you can see the lower band, it's getting used more. But that second doublet is also being used. Okay, so this is the drum roll, right? What happens in the presence of our mutations? What we see is in both doublets, there's a shift towards the UU. So that shift is not because of where it is. It's something about that sequence that the spliceosome is preferring more now. So, and it looks the same in PRCC. So weird. So if we separate out these two, just look at this ratio and this ratio, you know, in the wild type, it looks different. In the presence of our suppressors, they snap to very similar ratios. Super weird. We don't know what's going on yet, but it's very exciting. And so that's the third class. I'm going to skip that picture. So I've told you about these 4 dominant suppressors that promote an unusual splice site. Ha ha ha. I have the first evidence of functional role for PRCC and Kin17 in splice site choice. And part of what we think is happening is that these different phenotypes, these different ways that it can splice, suggest that there's multiple points in the splicing cycle where things are happening. So we actually think that PRCC and Kin17 are operating later on. You know, you've got that handoff, the handoff, the handoff. We think that this weird handoff is happening after those other steps. So yeah, I told you about the molecular machines and what I'm trying to do and what I've found so far, and a little bit about the world I live in now. So I'd like to thank my lab, who are awesome. Destiny's not in this picture, but she's been super helpful. I have collaborators and my committee, the university that I'm at, the Amamutsan tribal band, who represents the Waswas speaking people whose territory my university stands on, the funders who gave us money so that we can find out these answers to things. I want to thank the jellyfish, the bacteria, the worms who have been such good sports about all of this, and the international community of scientists who, through across borders, through fire and flood and pandemic, are working together to just try and understand how the world works. And so thanks to them, thanks to all of the collaborators, thanks to all the people who are interested in what we're doing. And, you know, I don't know, thanks to the world for being full of so many cool mysteries. All right, and I will take your questions.
Oh, awesome work. Does anybody have any questions for Jessie?
I'm gonna look in this chat.
Yeah, are there different spliceosomes that do different things, or is there one spliceosome that's just cutting everything?
Yeah. Oh, excellent question. So I was at a conference and I was learning all this stuff about spliceosomes, and at a certain point I was like, wait, it's not the spliceosome, it's a spliceosome. Every single one of them is likely to be kind of different. So there are parts that are on the machine in certain cell tissues that are just not present in other cell tissues. And so part of what's so amazing about it is that one gene can be used one way in a heart cell and used a different way in a muscle cell and used a different way in an eyeball and used a different way in something else. And so part of what might be happening is that there's some like, additional clip put on the machine so that in that cell type, it can splice everything correctly for that cell type. So it's amazing. It's very complex. I see, it's true he was on acid. Yeah, that's good times. I mean, that's, that's part of why I wanted to use the, I'm going to put this on gallery view so I can see anybody if they're still around with their cameras on. Yeah. Oh, Andrea, I thought there were more. Yeah, science. Heck yeah, there's so many amazing intersections between art and science, and it's amazing to see all the time. Yeah, cool. Oh gosh, I'm glad that you laughed at the pants joke because I laughed really hard, Siri. Thank you. Thank you, I appreciate that. Oh yeah, Toads, I can show you the animations. Yeah. Okay, I'll elaborate on the suppressors. Are they specific mutations or chemicals? Oh, okay. So that is a bit of, that is a bit of science jargon that snuck into my talk there. So when we say a suppressor, we mean something that like counteracts a mutation. So if the original mutation is the messed up splice site of UNC73, then a suppressor is a thing that changes the reaction. So each of those mutations that I found, the two mutations, in the class 1, class 2, class 3, all of those mutations are suppressors. So yeah, I don't know which Scott M that is, but that's a good question. How come the UU wild type splice site is? Christina, Parker, I never call you Christina, that was funny. Yeah, dude, what the heck? Why is that ever being recognized? I totally agree. It's super weird. It's super weird that it gets recognized in the wild type. It's already what a bizarre splice site this is. And then it's even weirder that these mutations push towards it. So some of the slides that I skipped is, the dress that's either blue and black or white and gold, how it's this like really ambiguous, like normally we all agree about colors, right? I'm like, this is orange, except for Al, who's colorblind, and so he just guesses. The dress is this wonderful example of an ambiguous color, and then people see it differently. So this splice site is an ambiguous splice site, and it just weird things are happening there. But that's cool because you can make discoveries there. I don't know if that explains your your question, that answers your question. The truth is, I don't know. I mean, give me more, what we say to the funders is give us more money and time, and we'll keep playing with all of our toys, and we'll eventually come up with an answer. Are these the same results found in normal worms, or is just in the wild types? So our laboratory version of normal is wild type. That's what we found. We didn't go out into the world and grab worms out of fruit, although there are labs that do that. There's a cool lab in France where they do that every year. They go apple picking and they pull out all these worms. And one time at a conference, I was talking to that PI and I said something about that these worms are soil dwellers. And she said, they're not soil dwellers, they're frugivores, in her thick French accent, which I cannot possibly replicate, because you don't find them in dirt. You find them in rotting fruit on the ground, which is just the cutest place to find worms. I mean, if you think of any other place to find them, it's way grosser. Yeah, so I don't know if that answers your question. RNA secondary structure. Oh shit, Parker coming through with the bio.
Not from me, by the way. They're from them. I don't know anything.
Okay. Yeah.
We got an enthusiastic audience over here.
Okay, yeah, good. I'm glad. Yay. Awesome. So there are, I mean, infinite complications to this story that I left out, most of which I don't even know, but a lot of the ones I do know I left out. And that is true that RNA can have structure, that it's one strand of nucleotides, and that it does, you know, in the DNA, there's the two of them and they're matched up. And so they're doing that, right? But in RNA, it's just one string. And so it's kind of available to fold up into weird shapes and do all kinds of weird stuff. So I did look at UNC73. I did a bunch of like structure predictors to try and see if I could find any kind of like secondary structure, if it looked like maybe that's why this was happening. And then I took them to Harry Noller, who is a professor in our department. He didn't get a Nobel Prize, but a lot of people feel he was robbed. Anyway, he's like 85 and he's super fun. And I showed them to him and I was like, do you buy this? Like these structure predictors, do you think it's real? And he's just like, you know, he knows the people who did all of this. He's been around so long that he's like, oh yes, I remember when Ted presented this, we thought it was going to solve everything, but it turns out that it's a much more complicated problem, you know, and then he's giving me all these details about it. And I'm just there like, oh, okay. Like, you know, these people personally, you've, you know, drank with them. I don't know. And he said he didn't really buy it in general. And so I could test it, and I may eventually. And see if that's what we think is happening. Even if it's not RNA structure, it could be something that's sitting down and kind of like blocking that spot, just kind of like occluding everything else. So that's another possibility too. But yeah, let's test it. Yeah, Nick, let's figure out what's going on. I know, I'd love to do some shape probing. Get it done in a couple weeks. That's amazing. Yeah, Harry Noller, he's a homie. He's pretty funny. I 10 out of 10 recommend chatting with him if you get a chance, because he'll tell you all kinds of silly stories about the 70s and the wild times in molecular bio. Cool.
Hey, Jessie, I have a question. So during your genetic screen, did you pick out anything that you just thought was super weird that you're like, how did you get in here?
You can hear all the banging, but my windows are all just like bang, 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 bang.
Where are you at?
I'm in Santa Cruz, but you have to imagine like a rickety old mansion, and then there's like a tower that sticks up, and like my room's at the top, and it's just like all windy and cold up here. Like that's what's going on.
So basically you're in an episode of Scooby-Doo.
Yeah. Yeah, it's very like that, except if somebody does laundry downstairs, the whole room vibrates. So I don't know if that's part of Scooby-Doo. Megan, what was your question? You're back online.
Sorry, yeah, I keep freezing. My question is, in your genetic screen, did you pick out anything that you just thought was super weird that you're just like, how did you pop out in my screen?
Well, honestly, the PRCC and the Kin17 is like, who the heck are you? Because those other ones that I showed, you know, that I kind of glossed through the class 1 and class 2. Those are the usual suspects. That's exactly who you expect to be a part of it. So if I was trying to figure out what part of the car is involved in steering, okay, yeah, I hit the steering wheel. That's not surprising, right? Like I would expect to get that. These two proteins are like out of left field. So Kin17 has been studied as a role in DNA damage repair. It hasn't really been thought of as a splicing thing. And PRCC, nobody knows what it does, but the most of the studying that's been done of it has been, as it's often found, as a cancer driver. So the first half of PRCC will get fused to another gene, because mistakes happen, you know, even the most well-managed library, come on. That'll get fused, and then that chimeric protein will go and just drive kidney cancer. And so nobody really knows what is PRCC doing normally. They only know what it does when it's doing that, you know, the Fantasia broom thing, where it's like, whoa, settle down, quit doing this, quit driving this cancer. So we've got these two proteins that are just like, whoa, where did you come from? So the thing that happened where we thought we kind of felt like we got scooped when we first saw it was that kin-17 got modeled into the spliceosome. And kind of basically the way our science works is that it's a back and forth between the structuralists who get the pictures of things and model things in, and us geneticists who test it and see where it looks, like what it does, right? So they say where it is, and then we say what it does, and we go back and forth. So if we identify something in our screen, then we literally tell our friends, hey, see if you can find some density for this protein. And the next time that they are putting together their models, if they find something that they're like, well, we don't know what this is over here, let's try that one that Al was just talking about, and they'll go ahead and see if it fits. And so there's this kind of back and forth. So, you know, as a geneticist, I'm always building on what the structuralists do. So they took a step ahead of me in that they found a piece of kin-17 in the spliceosome. And I was like, no, I'm the one who figured out that it was there. But they just found a little piece of it. And so, cool, this is always how it goes, you know, left foot, right foot, left foot, right foot. They figured out that it's in there. Now I'll tell you what it does and what it means. Although, of course, I don't know why it does what it does. I have no idea what's happening. I can carefully characterize what is happening. I don't know why it's happening yet. Yeah, we'll see. That's maybe another paper. That would be awesome. Ooh, Andrea.
Hi, Jessie.
Hi.
Quick question.
Ah.
So how, just curious, like how are those percentages calculated? Like when you say that this percent of the splice site is chosen?
Yeah.
Because like they're really close in some cases. Yeah.
Good question. So yeah, so this is part of what's so amazing about having Katiana around is that she does this stuff for me now. And Destiny, they super help me with it. So when you get those pieces of DNA, right, that the difference in their length, they're all the same on this end. So the difference in length is all from where they were cut on the other side, right? Where they were cut on the 5' side. And so you run them on a gel and they separate by length. And so we can tell from comparing it to the ladder that runs alongside it, we can tell that there's a band that's one nucleotide longer than the other band. So we do is we go into a computer program, we draw a little box around it, and we ask the computer, how much dark is in that box? And then the computer says, it's this much. And then we add together all the dark, and then we say, it's 12% of the dark in this lane is from that band, and 78% of the dark is from that band. So, you know, it's highly sophisticated.
So it's just off of like the pixels kind of.
Yeah, totally. Yeah,
Okay.
we'll just look at how dark they are. And the pixels are based on, yeah, Ewok science, totally. It's hilarious to me how it's such a combination of, you know, very sophisticated understanding and drawing together all of this information from all over, and then just some really, really simple things that work. And so we're like, cool. Yeah, and the dark that we see in the band comes from labeling. So we used to use radioactive labeling. Now we're using Psi3 labeling, which I hope means that when we all go back to the lab and we're all vaccinated someday far in the future, that means that we won't have a radioactive license anymore and we can drink coffee at our desks. That would be really nice if we could drink even just water. I just want to drink water at my desk. Mwah, we miss you, Andrea. I'm so glad to hear how you're doing. We'll catch up.
Oh, thank you.
Actually, in general, I miss all of you. And so it would be nice to be able to catch up and talk. I'm glad I got to see you here and tell you what I'm doing. And someday this will be over and we can like hang out like normal. I don't know. Yeah. One other thing that I think about the whole Ewok science thing is I remember early on when I had really switched to biology and I was starting to learn about this world of molecular machines, I was like, you know, we need some tiny scientists. We need some really small scientists because we are too big for this shit. Like how we can't see it, we can't touch it. It's like, how do we do this? And what I eventually realized is that these proteins, these enzymes, Cas9 and the restriction enzymes, those are like little tiny collaborators. Those are like this little tiny protein. And I'm like, hey, can you go in there and find all of these sequences and cut it? And this protein's like, yeah, sure, I got you. And then it just goes around and does it. And I just need to give it buffer, keep it happy, keep it its favorite temperature. And it goes around and does it. And then I'm like, thanks, man. I couldn't get to those. They were too small. So yeah, I really appreciate that. Thank you all for sticking around for so long and for coming and seeing my talk. I'm really happy. I had lots of fun making these slides, and I hope that if you already knew this stuff, I hope that it was a funny new way of explaining it. And if you didn't know it, then I hope that it makes more sense now. And then the next time that I'm telling you about what I'm doing, you can be like, yeah, this is that double thing that you're talking about or whatever. So yeah, yay.